A great day, everyone. We are the sixth group in Scientific Investigation Reporting. Accordingly, we are tasked to discuss the potentially hazardous biological agents, or PHBA rules, specifically sections 1 to 14, based on the International Rules for Pre-College Science Research, Guidelines for Science and Engineering Fairs. To give you a glimpse of how this discussion will flow, the fundamental purpose is to authorize the students to do projects involving PHBA, if and only if they meet the conditions beforehand. Indeed, this prominently ensures adherence to global biosafety regulations and guidelines. Ultimately, here are the 14 sections that aim to protect the students in times of experimentation.
Section 1, prior review and approval are required for the potentially hazardous microorganisms including bacteria, viruses, and others. Recombinant DNA technologies or human or animal fresh frozen tissues, blood, or body fluids. This section emphasizes the need for professionals to handle potentially hazardous organisms that can harm researchers if not taken to the proper extent. One of those needing administration is the rDNA tech, also known as recombinant DNA technologies, which conceivably sets issues such as gene pollution resulting from antibiotic-resistant microbes.
Section 2. An affiliated fair SRC, an IBC, or an IACUC must approve all research before experimentation begins. The initial risk assessment determined by the student researcher and adults supervising the project must be confirmed by the said committees. With the abbreviation stated, the first question that might come into our mind is, who are they? Fundamentally, Scientific Research Committee, Institutional Biosafety Committee, and Institutional Animal Care and Use Committee are the responsible committees that formulate policy, review projects involving biological materials like blood, semen, or urine, and approve researches that use animals in experiments respectively.
Section 3, experimentation involving the culturing of potentially hazardous biological agents, even BSL-1 organisms, is prohibited in a home environment. However, specimens may be collected at home if they are immediately transported to a laboratory with the BSL containment determined by the affiliated fair SRC. This section highlights biological safety levels, or BSL, that classify the levels based on the standards that must be enforced inside the laboratory. It is also stated that even the lowest level of BSL, which is level 1, must be controlled in institutions to competently manage the agents that may possibly harm human life and being.
Section 4. Research determined to be at biosafety level 1 must be conducted in a BSL-1 or higher laboratory. The research must be supervised by a qualified scientist or designated supervisor. The student must also be properly trained in standard microbiological practices. This section accentuates the need to experiment with laboratories if the BSL-1 group is identified. This contains biological agents that pose a low risk to personnel and the environment. These agents are unlikely to cause disease in humankind, animals, or plants, but preventive conventions and guidance are highly encouraged.
Section 5. Research determined to be a biosafety level 2 must be conducted in a laboratory rated BSL-2 or above, commonly limited to a regulated research institution. The research must be reviewed and approved by the Institutional Biosafety Committee if the regulated research institution requires the review. For a high school BSL-2 laboratory, the SRC must review and approve. A qualified scientist must supervise the research. BSL-2 risk group contains biological agents that pose a moderate risk. This is why further approval, supervision, and undertaking are needed to avoid severe infections or diseases.
Section 6. Students are prohibited from designing or participating in BSL-3 or BSL-4 research. BSL-3 risk group usually causes serious disease to humans, animals, or plants, resulting in vast economic consequences. The same thing goes with the BSL-4 group, but the effects are usually untreatable. Thus, students are prohibited from engaging in such circumstances.
Section 7. Laboratory studies designed to culture known clinically significant multi-drug-resistant organisms, or MDROs, must have a written justification for usage and be conducted at a regulated research institution laboratory with a minimum of BSL-2 containment and documented IBC review and approval. Representative examples include, but are not limited to, the following agents: MRSA, VISA or VRSA, VRE, CRE, ESBLs, and fungi with known resistance to antifungal agents.
Section 8. Insertion of antibiotic resistance markers for the clonal selection of bioengineered organisms is permitted, with the following exceptions being initially exempt from prior SRC approval, require at least BSL-2 containment. A. Students are prohibited from inserting antibiotic resistance traits or selecting organisms expressing traits that may affect the ability to provide effective treatment of infections acquired by humans, animals, or plants. B. Students are prohibited from designing or selecting multiple drug resistance organisms or MDROs to investigate the pathology, development, or treatment of antibiotic-resistant infections. This rule confers antibiotic resistance markers necessary for creating genetically modified organisms which the researchers can utilize to select the new gene's insertion. As a result, this system requires at least biosafety level 2 containment. Additionally, this also prohibits students from selecting MDROs to examine the traits of antibiotic-resistant infections. Alarmingly, these bacteria resist treatment with more than one antibiotic drug, and the more often the antibiotics are used, the more likely the bacteria will develop resistance.
Section 9. Extreme caution must be exercised when selecting and subculturing antibiotic-resistant organisms. Studies using such organisms, including BSL-1 organisms that may have been exempt from prior SRC approval, require at least BSL-2 containment. Students who undergo such experiments are strictly warned to treat antibiotic-resistant organisms as potential pathogens. Hence, at least biosafety level 2 must be administered to limit the risk that may take place in the duration of the investigation.
Section 10. The culturing of human or animal waste, including sewage sludge, is considered a BSL-2 study. This section justifies the organic fecal material that comprises a variety of pathogens, bacteria, viruses, and parasites. Thus, a BSL-2 must be contained as it poses a moderate risk to the human environment.
Section 11. Naturally occurring plant pathogens may be studied at home but may not be introduced into a home or garden environment. This section upholds that pathogens found in plants can still be detrimental if not duly contained. Therefore, introducing such biological aspects in our home or gardens is prohibited and not advisable by health standards.
Section 12. All potentially hazardous biological agents must be properly disposed of at the end of experimentation under their biosafety level. For BSL-1 or BSL-2 organisms, autoclave at 121 degrees Celsius for 20 minutes. Use a 10% bleach solution. Incineration, alkaline hydrolysis, biosafety pickup, and other manufacturer recommendations are acceptable. This rule is applied with the risk categorized by biosafety levels. Thus, laboratories must have the capacity to decontaminate waste by having pieces of equipment or other methods which can be used substantially.
Section 13. Any proposed changes in the research plan or project summary by the student after initial local or affiliated fair, SRC approval must undergo subsequent SRC or IBC review and approval before such changes are made and before experimentation resumes. Looking back into Section 2, these committees are knowledgeable about the regulations concerning biological agents. That is why the roles of SRC and IBC are vital before conducting the methodology of a project.
Section 14. The following forms are required. A. Checklist for adult sponsor or Form 1, Student Checklist Form 1A, Research Plan or Project Summary, and Approval Form or Form 1B. B. Regulated Research Institution Form or Form 1C when applicable, and C. Qualified Scientist Form or Form 2 only when applicable.
And that concludes the 14 sections under the PHBA rules. We hope that this video may help the proponents succeed in their vision towards a new era of science and technology where good governance rules society. Thank you for listening.